Hello everyone, this is Dr. K. SIVASUBRAMANI, Department of Microbiology, ANNAMALAI University. We are going to see something about microbiology in the era of metagenomics. Microbiology has evolved a lot from the plate period to with the development of molecular biology, microbiology has reached new heights. Basically, what is genomics? Genomics is a discipline in genetics that applies recombinant DNA technology, DNA sequencing methods, bioinformatics and sequencing and a lot. And proteomics is the study of the entire amount of proteins in a cell. And metagenomics. Metagenomics is a very modern science which can be defined as the genomic analysis of the collective microbial assemblages found in environmental samples. When we analyze environmental samples like soil samples, usually when we take even 1 gram of sediment sample, it can contain 10 billions of cells. It may contain 4,000 different species, and it can have a huge length of around 1,500 kilometers of DNA. But when we try to culture these samples on plates, we get very little amount of isolates only. Very little bit of isolates only. This is known as the great plate count anomaly. Why it is happening? Because in environment, microbes are living in a very different condition. The pH is different, oxygen condition is different, and due to other environmental conditions, different organisms are thriving in environment. When we try to culture those organisms on Petri plates, we cannot provide the exact environment to the microorganisms. That's why we are able to isolate only 1 to 2% of organisms on Petri plates. Suppose we have isolated a lot of enzymes, lot of antibiotics and lot of other metabolites from these small group of organisms. What we can isolate from the remaining 98% of organisms? That's what we are going to do with metagenomics. Using metagenomics, we can isolate the potential benefits from other remaining 98% of organisms. Metagenomics can be done using different methods. Either the first thing in metagenomics is we have to isolate the DNA from the entire organisms, and then we can either go for sequencing method or we can go for library construction method as displayed in the image. Where we can use this metagenomics? We can use the metagenomics in life science. We can use the metagenomics in medicine, like the analysis of human microbiome. We can use metagenomics in earth science. We can use metagenomics as a source of alternate energy. When the environment is polluted, we can use metagenomics as a remedy for environmental remediation. We can use metagenomics in biotechnology. We can use metagenomics in agriculture. We can use metagenomics in biodefense and microbial forensics too. Still, as a developing field, metagenomics has some limitations. When we go for sequencing, we produce too much of data. Too much of data, it means we are unable to use the benefits from the data. And the contamination, chimeric clone sequencing, these are other problems. And another important thing is the method is subject to extraction biases. So these are some limitations of metagenomics, and we are going to see some interesting cases about metagenomics. And this gentleman is Craig Venter. He is well known for his endeavor in contribution in human genome sequencing. His other contributions are great ocean sampling and in synthetic biology. He went for